Hal.
Hello, everybody. Can you hear me?
Yes.
Yes.
Good. To say the least, this has been a very unusual year with COVID changing all aspects of our lives, but with the hope that vaccines can bring us back to some bit of normalcy. We are privileged today to have Joel Ernst here, who is a world-class expert on vaccines and a professor and director of experimental medicine at UCSF, to give us a talk on the latest news on COVID vaccines. This obviously is a timely and important topic for everybody, and I think there will be lots of questions. So Joel has graciously agreed to keep his talk to 15 minutes and then allow 15 minutes for questions. The questions can occur in real time by taking yourself off of mute or using the chat room. The title of Joel's talk is Science and Art of COVID Vaccines. Joel, are you here?
I am.
Oh,
Thank
that's a
you.
good thing.
Hello, everyone, and thank you, Hal, for the opportunity to speak with you again. As you mentioned, things have changed a lot since our last encounter. I'm going to share my screen here. And I'm going to talk about vaccines for sure, but I'm also going to talk about kind of another timely topic, and that is the fact that we're now starting to see some variation in the SARS-CoV-2 virus. So I've got 15 minutes. I'm going to evoke my 15 years of working in New York City and speak like I come from New York City and get this done. So first of all, as you're surely aware, there are two authorized COVID-19 vaccines available right now in the United States. And I'll refer to them by the names of their sponsors. One is the Pfizer-BioNTech, and the other is the Moderna. These are the two concerns that have developed and brought them into the clinical use.
Excuse me, Dr. Ernst, would it be
Yes.
possible for you to put that in full screen to go into?
Yeah, sure. Absolutely.
Thank you very much.
Thank you for reminding me.
Yeah.
Yeah, you didn't need to look at all that other junk on my desktop. The vaccines are actually very similar. They are both RNA-based vaccines, so they have the RNA that codes for the so-called spike protein of the SARS virus. And since RNA is a fragile molecule, it's incorporated into something called lipid nanoparticles. And both of them have that same basic format. Now, it's notable that up until now, there have been no RNA vaccines previously authorized or approved for human use. So this was a really accelerated development program. That said, there was a lot of background preclinical work that was done that gave people some confidence that this would be safe and actually would work. It's probably come to your attention that this is the most rapid vaccine development in history by a long way. Up until now, the previous record was about 4 years, and we're talking now about 10 months. Some people have found that a source for concern, but I actually would try to allay that concern by saying that there were really no steps that were skipped in the process of developing these vaccines or in testing them. But rather what happened was that the different steps were done in parallel and not in series. In other words, the standard way to do it is, let's make this vaccine, let's test it in animals, then let's test it for safety, and then let's do a rollout and test it for efficacy. And if it seems like it works, then we'll make lots of doses of it and manufacture it at scale. Instead, the manufacturing development process was really done in parallel. So basically, these were made in millions of doses, tens of millions of doses before anybody knew that they were going to work. So that was de-risked by the Operation Warp Speed funding. And so it's really important to say that there were no steps skipped in evaluating the safety. So the side effects of both of the vaccines are fairly common. They're local pain, fatigue, kind of feeling wiped out, having a headache, muscle pain. Those are more common and somewhat more severe after the second dose in some people. But there's an interesting observation, and that is that the side effects are actually less common and less severe in people older than 55 or 65. Why that is the case, no one can do anything but speculate, but it's a pretty durable observation with both of these two vaccines. The side effects are transient. They last about 24 hours, and the muscle aches, the fever, if somebody has a fever, the pain, pretty much respond to Tylenol or Advil, that sort of thing. With respect to other aspects of safety, there were some transient changes in blood cell counts that probably reflected the immune response to the vaccine, but no changes in blood chemistry makeup. And there have been, as I'm sure you've heard, 3 results of severe allergic reactions. It's interesting that those happened very shortly after the vaccines were released for public use, but they actually haven't come back. In other words, there haven't been more examples. There are now up to, there are probably 3, there's possibly a 4th one. But this is not something that we're seeing commonly. Now, I thought I'd put this plot up for the 2 and explain it, because I think it really makes a dramatic point in both cases. So what these are plots of the incidence of infection. In other words, new infections on the y-axis on the left side there, and then across the bottom is the time after the first dose of vaccine. And on the left is the Pfizer-BioNTech data. On the right is the Moderna data. And what you see here is that the people who got the placebo in both cases basically were progressively exposed and got progressively infected. They developed symptoms, they got tested, they had confirmed infection. And this happened in the placebo group, obviously, with both. The curves are slightly different because the populations are a little bit different. But what you see here and here is that within about 2 weeks after the first dose, this vaccine recipient curve just flattens. In other words, within a couple of weeks of the first dose, the immune response is kicked in. It's an effective immune response. And already at that point, you see these curves separating so that the people in the control group keep getting infected and the people in the vaccine recipient groups do not. And the efficacy was very, very close. It was 95% for Pfizer and 93% in this form of the data analysis from Moderna. I think those are indistinguishable. I wouldn't sort of place a nickel on thinking that one of them is better than the other. So that's the efficacy in the whole group. And then I think a lot of people are interested in, yes, but sometimes a vaccine works in one group, but not another. But the good news here is I've just put the percent protection, the percent efficacy for each of them here in bold. The overall, as I mentioned, was 93 or 95, depending on how you calculate it. But there was no drop off with age. In other words, for older adults, the vaccines were just as effective as they were in younger adults. They were equivalent in men and women. And in this slide, they were essentially equivalent in ethnically and genetically diverse people. In other words, Blacks, Hispanic people, and I'm not quite sure how communities of color were otherwise identified or defined. It's a little bit vague in the FDA submissions. And so ethnic diversity did not seem to be associated with any difference in protection as well. So with respect to age, with respect to ethnicity, the vaccines both seem to be equally efficacious across the board. Now, the other question was, what about people who get infected despite being vaccinated? Do the vaccines have an impact on the severity of illness? And it turns out that they do. So the numbers are very small here, so I wouldn't make too much out of the absolute numbers or the percentages at this point. But of the people who received either placebo or one of the vaccines, there was a certain incidence of severe COVID-19. In other words, COVID-19 disease that required hospitalization. And in the vaccine recipients, those were dramatically reduced in frequency. And so I think it's safe to conclude that both vaccines not only prevent symptomatic infection, but they prevent or markedly diminish the likelihood of progressing to severe disease. So just with respect to conclusions on the vaccines, both of these RNA vaccines are highly efficacious. They have similar and I'd say acceptable side effect profiles. There's really little difference in their efficacy by age, sex, or ethnicity or race. However, we don't have any data yet on their safety or efficacy in children, in pregnant or breastfeeding women. The data on children are in the process of being generated now. With respect to pregnancy and lactation, the CDC's recommendation, which I think is reasonable, is that if a woman is in a high risk group, either high risk for becoming infected or exposed, or high risk for a bad outcome, even if they are pregnant or breastfeeding, they should be vaccinated. And then finally, there's no real data on transmission. In other words, on a population level, you can imagine that by reducing the frequency of infection, you'll reduce transmission. However, in the people who've been vaccinated that for some reason become infected anyway, we don't know whether the vaccine alters their likelihood of transmitting infection to other people. So that's what I have prepared on vaccines. I just want to take a few minutes then to talk about the new viral variants because they've really been making the headlines now for 2 or 3 weeks. So there's one major variant that's being much discussed, and it's a variant that was first identified in southeastern England, I think it was. And there was a concern because it was spreading rapidly in England. It's now in the United States. At least 33 states have identified cases, and it's pretty likely that it's emerged elsewhere. And the main point about this variant is that it has multiple mutations. None of those mutations seem to be associated with more severe disease, but they do seem to be associated with a higher frequency of transmission. In other words, a given person is more likely to transmit this virus to additional other people than of the earlier sort of parental version of the virus. It's widespread, and I think that the widespread nature of it is because it's more transmissible. It's growing in frequency everywhere it has appeared. There is substantial reason to be concerned that this will lead to worsening of the surges we're already enduring and the surges we may be looking at in the near future. As I mentioned, the mutant is not associated with increased severity. There is evidence so far that the vaccine recipients from the original version of the spike antigen are still protected against infection with this variant. So it has not escaped immunity. And so really the point of it all is until everyone is vaccinated, we have to continue masking and social distancing. And so that's what I have prepared, and I've hopefully left time for a sufficient number of questions.
Yes, we actually have a lot of questions. Thank you very much for that. So let's see, Megan started this. For the vaccines that require 2 doses, do both have to be from the same manufacturer?
That's a great question. The easiest dodge is to say no one's done that experiment. No one would probably recommend doing that by choice. The administration schemes are pretty well set up so that when somebody gets one vaccine, the interval between the first and the second vaccines is different. It's 4 weeks for the Pfizer and 3 weeks for the Moderna. If there is a supply shortage when somebody's due for the second dose, then I don't think it would be unreasonable to be vaccinated with the other because these 2 vaccines are really quite similar.
Okay. Can one choose a vaccine from the 2 choices offered?
It depends on where you are. Most places, no. Most places give you the vaccine that they have, and the supply is obviously finite and changing. And so I don't know of any place that's actually given people the choice. However, we had sort of acute situation at my hospital because we were administering vaccine at two different sites on a campus. One was giving Moderna and one was given Pfizer. And after the first day, everybody knew which site was giving which vaccines, so they could figure out where they wanted to line up. Um, but officially, no, you're not supposed to be able to choose.
Okay. Why are we hearing that so many nurses and physicians are hesitant to get the vaccine?
You know, that's an unfortunate situation. I think there's certainly there's certainly the vaccine opposition sentiment. There's vaccine hesitancy as well. And I think a certain fraction, I think, of doctors and nurses are aware that there are side effects to just about everything we do in medicine. And they may be a little bit wary of the side effects of the vaccine. But certainly, the side effects of the vaccines are certainly less severe than the side effects of getting COVID. So I think if somebody's hesitant to get the vaccine, they want to kind of wait a little while and see if everybody else is okay first. Um, I can understand that, but I would say that people that are at substantial risk and on the front lines seeing patients really ought to think twice about deferring vaccination. They should probably get vaccinated as soon as possible.
Has any testing of youth been done, and when would the vaccine be available to children younger than 18 or 12?
So the main reason that that children weren't included in the trials was because of early evidence that children were less susceptible. Now we know that children are susceptible, but they do seem to be less susceptible to severe disease. They're also less likely to transmit it. So if you think about the trials from kind of a perverse angle, the trial designs benefited from having lots of infections so that you could compare the vaccine and the placebo very quickly. And that's one of the reasons they got approved so quickly. So because of the issue with children, children were excluded. Children are now included in new phase 3 trials. I think that the speed with which those will reach endpoints is going to be determined by the frequency of infection in their communities. I think we'll get safety data fairly quickly, and I think that's probably the main question. In other words, is there any reason to think that these are not safe in children? And so far, I'm not aware of any side effects in children that have appeared that haven't appeared in adults.
The children that are being tested, how young does it go right now?
So the trials that I'm aware of are 5 to 12 years old. So very young children still not included.
Regarding transmission of disease by vaccinated people, are there other diseases where people who are vaccinated still transmit the disease?
There are a few examples, but they are mostly in the case of vaccines that are less efficacious than this. What we don't have a lot of data on so far is it seems like sort of a detail at first. So there are vaccines that prevent disease, and there are vaccines that prevent infection. Now, the difference is a vaccine that prevents only disease can still allow infection to happen, but the person just doesn't get sick. And we actually have a fair number of vaccines like that. This is one of the things that measles vaccine does. It's a great vaccine, but it actually doesn't provide protection against infection. It prevents disease. So if we have a situation like that, and it's analogous with COVID, and we don't have the data one way or another yet, it's certainly theoretically possible that somebody can be protected from getting symptomatic infection, but they still might become symptomatic disease. They still might become infected, and they still might be able to shed virus and transmit to other people. So more information is needed to really know the answer to that.
Can you speak about the AstraZeneca Oxford vaccine from the UK?
I can. So that is actually a different category of vaccine. That is what's called an adenoviral vectored vaccine. So An adenovirus, which ordinarily is part of a family of viruses that causes common colds and upper respiratory infections. This version is crippled so that it can't replicate, it can't multiply in the body. And it's modified to stitch in the spike glycoprotein from the SARS virus, SARS-CoV-2 virus. And so it's also injected. It's also injected in 2 doses. And the initial clinical trial was really actually unfortunately complicated by an unfortunate dose issue, which gave a surprise result, and that is the people who got the lower dose for their first injection seemed to be better protected than people who got the higher dose that was the planned higher dose. People are trying to make sense out of that, but it's a little bit confusing then to decide, well, how effective is that vaccine really? In other words, that vaccine gave a lower efficacy than either of the RNA vaccines when it was given according to the planned doses. It seemed more effective in the people who got the lower first dose. Now, whether that's a statistical quirk, whether that's because more people that got the lower dose were younger or less exposed, remains to be seen. So I think they're really having to regroup and redesign their phase 3 trial in order to get a firm notion of where it fits into the larger picture.
What is the relationship of infected, the number of infected, and probability of variants?
So the virus makes random errors. In other words, when it's replicating its RNA, it makes random mistakes. That's just part of biology. That's always happening. RNA viruses are a little bit more prone to doing that. This RNA virus actually makes fewer mistakes than other RNA viruses. So it's a random process. And those mutants then get spread, and some of them are successful and some of them are not successful. Some of them are crippled and can't cause disease. So obviously, the more infections there are, the more viral particles there are, more opportunities for errors to be made, and more opportunities for those error-riddled viruses, mutant viruses, to just try out and see what works. So obviously, the higher fraction of people that are infected and transmitting, the higher likelihood of variation. And so masks, physical distancing, hopefully vaccination will help subdue the number of people infected and transmitting, and so lower the frequency of mutation and variation.
What is your opinion on one shot to get more people vaccinated versus everyone getting 2 shots?
Good job.
It's a great question, and I'm glad that you framed it with what is my opinion, because there are really no data. We can kind of try to extrapolate from data, but my opinion is based on the following. The clinical trials were designed and executed the way they were with 2 doses. Those are the data that we have. Any deviation from that routine, we don't know what the impact is going to be. We knew a little bit about how many people got infected between the first dose and the second dose, and it was fewer in the vaccine recipients in both cases than in the placebo recipients. That doesn't tell us at all how long their immunity will last, and it also really doesn't say whether they're protected as well. So I think the other issue is here in the United States, we're having trouble getting vaccines into people. There are more vaccines sitting in freezers than we have getting into people's arms. And so I think until we solve that problem, it's really impractical to talk about trying to vaccinate twice as many people when we can't even vaccinate the people that, we can't even administer the vaccines that we have in stock.
Can you speak more to the evidence that the vaccine is still effective against the new variant?
I can. So the experiments, or the way that those studies were done, were to take an example of the variant virus and serum samples, blood samples from people who'd received the Pfizer BioNTech vaccine and just studied the ability of the antibodies in those vaccine recipient serum to block infection of cells in culture and compare the ability of their antibodies to block infection by the earlier, I'll call it the ancestral version of the virus, compared with the variant. And there was a slight decrease in the ability to block the variant, but it really was not it wasn't a difference that was probably biologically important. So it was a bit of a small study. It's the kind of study you can do fairly quickly and get results out. This just came back out day before yesterday, I think it was. But so far, it looks as though the vaccines protect against this new variant.
Can you speak to what appears to be a poor or helter-skelter distribution of the vaccines in California?
Not without getting too political. Frankly, it has been patchy everywhere. And I think we're not any worse in California than we are, across the United States. And I think it's partly the fact that we're trying to do it quickly and didn't have a lot of time for planning. It's partly because our public health infrastructure has been weakened over the years by budget cuts. We know it's possible. We know that we're going to have to get creative and start thinking about vaccinating people in non-traditional places. I'll just sort of recall that in 1947, there was a small outbreak of smallpox in New York City. 6 million people were vaccinated in a month. And so that's what's possible. That was what was possible because there was a very effective director of public health who had politicians' support and basically knew how to speak to the public and get people vaccinated. So I think it's all possible. We need to get more creative. We need to start giving vaccines in, idle football stadiums and basketball stadiums and things like that, and not expect people to go to clinics or pharmacies.
Have you received the vaccine?
I have.
Right.
Right here, last Thursday.
This one I don't quite understand. Avis, this is you. I have heard some have had mental issues. Can you speak to this?
So the mental issues that I've been aware of are from infection, not from vaccination. And there is a certain frequency of confusion, memory loss, inability to concentrate. They have been associated with infection, and I have not heard of any examples of that occurring with vaccination. It's not terribly uncommon for severe illness and severe infectious diseases to have that kind of impact on people. So I don't think I found it terribly surprising.
Is Pfizer's vaccination riskier since it has to be frozen?
There's no evidence for that. They both do need to be frozen. It's just that the Moderna seems to be a little bit more stable outside the freezer for a little bit longer. As long as they're handled appropriately, they should be equally safe and equally effective.
When someone tests positive, they are told to stay home for 10 days. If they have no symptoms to begin with, is a 10-day quarantine really long enough? How do we know?
So that's a really excellent question. And it's been the source of substantial frustration for people. It was more frustrating when it was much harder to get tested. And so now I think there are emerging policies that have to do with being tested after exposure, retested or tested 7 days later, and having a shorter quarantine period. But the main reason for that quarantine period, there are really two main reasons. One is the incubation time, the time between the time somebody was exposed and the time that they actually develop symptoms can be as long as 2 weeks. And second, we certainly know that about 50% of the cases of COVID in the United States were transmitted by asymptomatic people. This is probably the main hazard of this virus, is that it is transmitted by people who don't even consider themselves to be sick. And so the quarantine period is really meant to account for that combination of things. And as frustrating as it can be and as difficult as it can be, it's really an important element of trying to keep things from becoming worse out of hand.
Okay. Does the new RNA technique have possibilities for developing vaccines for Ebola, AIDS, and other serious viral diseases without vaccines to date?
That is also a great question. In fact, one of the early RNA vaccines was for Ebola. It didn't progress quite as quickly, and it actually ended up getting sort of bypassed by another Ebola vaccine that turns out to work very, very well. And so it probably isn't going to go very far for Ebola, except under one circumstance, and that is the existing Ebola vaccine is only against one or actually three variants. If an Ebola strain emerges someplace that doesn't fit that family, then we're going to need a new Ebola vaccine very quickly. And one of the advantages of the RNA vaccines, and one of the reasons that are the first two we have available, is that they can be modified very quickly. In other words, you can make a new version of the RNA literally overnight. So with respect to HIV, the challenges are different because one of the things that is so challenging for HIV vaccine development is the massive number of mutations that it undergoes. And so as soon as you've got an antibody to one version, it can develop an escape mutant to escape that antibody. There are a small number of antibodies that are so-called broadly neutralizing. We haven't figured out how to generate those yet. If an RNA vaccine strategy is advantageous for that, then it certainly could help solve the HIV vaccine challenge.
Okay. Dr. Ernst, we have several more questions, but I'm conscious of the time limits of our members. So if you wouldn't mind staying to answer the questions, I'll go ahead and shut down the meeting. And if people want to leave, they can leave. And then if you want to answer the questions, that'd be great. Okay.
I can stay for about another 10 minutes.
Okay. Let me go quickly then, quickly as I can. Oop, wrong button there. All right. Let's see. Okay, same as we did last time with you. We did donate in your name to the End Polio. It is a 2-for-1 matching with the Bill and Melinda Gates Foundation. So with that donation, we will inoculate 150 children. Next week, we have a club assembly, if I have this correct, on the Wine Fest, how to bid on auction items. Quote for the week, it is our attitude towards events, not events themselves, which we can control. Nothing is by its own nature calamitous. Even death is terrible only if we fear it. Okay, congratulations. And Jana, I didn't point you out on your baby picture too, but we did see it. So thank you very much. Welcome to Los Gatos Rotary. And thank you also our visitors. So Ken, Terry, Jeffrey, I saw Barbara Fishman on there, Jay, Karen, and our good friend Charlie Erickson. Thank you for playing today. Somehow I've screwed up my thing. So with that, stay safe, stay healthy, stay connected. Thank you all. Meeting's adjourned. If you want to drop off, you can. Otherwise, we will go back to Dr. Ernst.
Okay.
And let's see. How long will vaccine immunity last?
We don't know yet, but it's looking pretty promising. So needless to say, with such a new vaccine, we don't have years of follow-up to know. But what's been done so far is to just measure the stability of the antibodies that develop in response to the vaccine. And right now we've got data that go out 4 months where there's really no significant drop off. And so if you extrapolate those curves out, it looks like we should have several years of immunity. Um,
With hospital ICU space so low, will opening schools, even with low risk, add to the community risk?
Uh, that's a little bit outside my field of expertise. That's really a kind of epidemiology, mathematical modeling question. Um, I think what we know is that school age children don't transmit very efficiently to other school age children. The risks are actually transmitting to teachers and teachers to teachers. I think that that is one of the reasons that teachers are among the next priority groups for vaccination, so that that can really go on. And that'll be done obviously in parallel with vaccine trials in school age children.
Okay. What is the limit of time for someone to get a second dose for it to be equally effective?
No data on that. Um, so in the absence of data, I can speculate just a little bit. I mean, the clinical trials allowed a 3-day window before or after the designated 21 or 28 days. And that's kind of just a reality of doing clinical trials. And so there were not a lot of protocol deviations that allowed anybody to make a conclusion about what's the, what to do if it's 6 weeks instead of 3 weeks or 4 weeks. I think in the absence of data, kind of knowing about the basic principles of immunologic memory, I think it's probably fair to say that you can wait a month or two without much damage. But I think after that, then in many cases, if a vaccine needs to be given in multiple doses and a booster is done late, sometimes you have to start from scratch and start over.
Okay. If one already had COVID-19 and they have the antibodies, does this safeguard them from getting the virus?
It so far the data say that having had COVID, recovered, and having antibodies does protect against reinfection. However, most people who had COVID and recovered don't get tested for their antibodies. And so the recommendation actually is if your infection is more than 90 days in the past, then you should be vaccinated if you're in an otherwise, vaccine appropriate group. Um, and there's no evidence for harm in doing that, by the way.
Okay. The world is developing scores of vaccines. Will we see any of those here in the United States?
I think we're probably going to see the AstraZeneca vaccine, but it's going to be a while yet. I think the other vaccine that we'll probably be seeing in the United States is the one from a company called Novavax. And the Novavax vaccine is based a little bit like our present tetanus vaccines or tetanus or diphtheria vaccines. In other words, it's the spike protein plus an additional substance called an adjuvant to kind of wake the immune system up and tell it that there's this foreign antigen to respond to. In preclinical studies, the Novavax vaccine looks very promising. Their phase 3 trial is just beginning. So I think those are the two next ones that we're likely to see in the United States.
Okay. And then is there anything known about long-term effects of the vaccine 3 to 5 years out, and how will we know?
We can't really know that because these vaccines were only put into humans beginning in May of this past year. There are some reasons that I feel reassured that there shouldn't be substantial long-term risks, and they have to do with the composition of these vaccines. So RNA is a fragile molecule. I mean, it literally falls apart if you look at it wrong. And there are enzymes all over the place, literally everywhere, on specks of dust, enzymes that will degrade RNA. So the RNA is not going to persist. The so-called lipid nanoparticles are made of lipids that we know a lot about how they're metabolized. We know that they are metabolized, so they don't persist in the body. They can't travel around and kind of affect distant organs and things like that. So I will be very, very shocked if there are long-term complications of either of these.
What are current conditions at hospitals regarding capacity?
Boy, it depends on where you are. Obviously, Southern California is really catastrophic. Here in San Francisco, we've been setting daily records for a number of people in hospitals with COVID. However, we're also accepting some patients from Southern California. In other words, where the capacity is exceeded, we've been taking in transfer patients. We did that with the earlier surge in the fall. It's being executed a little bit better this time because we took some patients from Imperial County back in the fall that were airlifted to San Francisco, brought by ambulance to our hospital, and nobody thought about what to do when it was time to send them home. We're better at it this time. They're getting to go home. And so San Francisco is certainly seeing a surge. The surge isn't as serious as it is in Southern California. I'm not so aware of what the situation is like in San Mateo or Santa Clara counties right now.
Okay.
Do we have time for just a quick verbal question?
Yeah, go for it.
I didn't know how to word it. It's actually not a vaccine question. My question is, if I am infected by somebody that it maybe is in the hospital and they're quite severe versus I'm infected by somebody that is asymptomatic, is that really, I mean, is there a difference, or is it basically your own human body that just reacts differently?
That is really a great question. I'm a little bit surprised it hasn't been asked more often because it's really important. It's mostly about the individual's response to the infection rather than a virus that causes more severe disease or less severe disease. There is a little bit of a modification because if there's, and we don't know that this is so yet, if there's a difference in the number of viruses shed and the number of viruses that somebody gets infected with, we think that probably is associated with a more severe outcome. But it's hard to study that. It's hard to prove it or disprove it. It's just sort of a hunch that most experts have. But right now, the precautions are basically the same. And obviously, the precautions, for example, for nurses in intensive care units are especially high, not so much because they're exposed to more virus. They're just exposed to it for longer and by more patients. So the answer to your question is probably it doesn't make much difference.
Okay. And keeping in line with your Schedule 2, one more question. Why do the elderly have less side effects than younger people?
Just lucky. It's really, it's exactly, it's really interesting because obviously that's not the case for everything by any means. In fact, it's kind of a reverse in many cases. But it's really a pretty durable observation. I'd say don't question it, just take it. Just take advantage of it.
We will.
All right. Well, thank you very much for your time and your presentation. It was
Happy to do it. And thank you for the donation for the poliovirus campaign. This is a topic near and dear to my heart. So it's important to me, and it was really gratifying to hear that. Thank you.
Thank you, Joel, for that superb presentation.
Definitely.
Happy to do it. Take care, everyone. Stay well.
All right. You too. Thank you.
All right.